Yeah, thanks to the organizers for giving us the opportunity to present our work here today. So we are experiencing this current push towards increasingly high-dimensional neural data. We now have access to recordings of tens of thousands of neurons in behaving animals over long time scales. And a view that has been key towards interpreting this data is that of the state space, where the state of the system at a particular instant in time can be viewed as a vector in a very high-dimensional state space, where each axis corresponds to the activity of one neuron. And as time progresses forward, the state of the system changes. And what has been argued is that although this state space is very high-dimensional, the state of the system is often constrained to some low-dimensional submanifold of the state space. For example, here a circle. And manifolds are ubiquitous in biological data. So for example, in systems neuroscience, it has been suggested that the position of the head of an animal is encoded along a circular manifold. If you are more interested in cognitive variables, more abstract features of images are encoded on a planar manifold in the prefrontal cortex. And even beyond neural data, behavioral data can be encoded on some low-dimensional manifolds. For example, the position of the leg of a Drosophila as it walks has been suggested to be constrained to some cylinder. So what all these examples have in common is that there is some underlying dynamical system that generated the data. And in this work, we are asking what are the dynamical system's principles that underlie those manifolds. And first, we argue that dynamical systems can be manifold generating. So many dynamical systems in biology can be described in terms of a differential equation where the F describes the interaction between the variables that you are observing and the U describes the inputs to the system. And already there, different properties of this F can generate different manifolds. So if the vector field defined by this F is periodic, the manifold, the trajectory of the dynamical system will be that circle. And if it is aperiodic, it will be a line. And we can gain similar insights into higher dimensional manifolds. Similarly, looking at the properties of the input to the dynamical system can provide insights into the manifold of the system. So what we show is that if the inputs are constrained to some low-dimensional manifold, where different points on this manifold correspond to different time-varying input to the system, then what we show is that the state of the system must be constrained to a low-dimensional manifold too. And furthermore, there are some constraints on the topology of the manifold of the state of the dynamical system that come from the topology of the manifold of inputs. So for example, one direction on the manifold of this state of the dynamical system will encode for time, and one direction will encode for the input identity. And so we've split these two works into two posters, one that's presented today and one that's presented tomorrow. So now I'm going to very briefly give you insight into what we can tell about particular datasets using this link between geometry and dynamics. So first on behavioral manifolds, I've briefly alluded to that previous work has suggested that the position of the leg of a Drosophila as it walks is encoded along a cylinder. And using a combination of data analysis and modeling, we've shown that more than a cylinder, the position of the leg of the Drosophila is better described as living on a torus. And the reason that we are seeing a cylinder in the data is that we only have access to a small range of speeds. So we only have access to a cut of this torus corresponding to this range of speed that we are observing in the data. So in neural manifolds, we looked at recordings of the visual cortex as an animal is looking at gratings of different angles. And the experimentalist recorded the same neurons through an entire month of learning some task that depends on these angles. And what we've shown is that over learning, this manifold that encodes the grating angles has a stable shape, so its shape doesn't change, but the dynamics that generate the manifold do. And finally, on RNN manifolds, so we've considered a bunch of RNN tasks, but in particular this working memory task where we're feeding a sequence of two inputs to an RNN. It has to remember it through a delay and then produce the same sequence of outputs. And what we've shown mathematically is that the state of the RNN at a particular instant in time must lie on the torus. Furthermore, at the time of the first output, the great axis of the torus encodes for the first variable that must be outputted, such that the projection of this torus on the decoder subspace is the circle that corresponds to that first output. And as time progresses, the geometry, the intrinsic geometry of the torus dynamically warps, such that now, at the time of the second output, it is the second variable that is encoded along the great axis of the torus. So with that, I would like to thank my collaborators, and in particular, Isabel Cornacchia, who was supposed to present here today, but couldn't come. Thank you.
Thank you so much, Rajat. I would invite our next speaker. Is Philipp Werthmann from Institute for Neuromodulation in Paris.
My talk, or my poster, is about estimating communication subspaces with low-rank RNNs. It's supposed to be a follow-up of Joao's talk. Maybe then he will give a follow-up on the same work tomorrow. Yeah, exactly. So we're interested in finding structure in across-region interactions and have some good insight already from this influential paper named Cortical Areas Interact Through a Communication Subspace, where they have simultaneous recordings from two brain regions, namely V1 and V2, and then reduce or subtract the stimulus signal from the neuronal traces and study the trial variability or noise correlation. They then apply a reduced rank regression and find that V1 communicates with V2 through a lower dimensional subspace. We would now like to expand this paradigm and study signal correlation or condition variability with a task, we just call it the MANTE task, a contextual decision-making task. And we also have parallel recordings of 6 brain regions. This makes it a little more challenging, so we're using multi-region lowering RNNs. I will introduce them now. So let's start with a vanilla RNN. In our case, we're mapping neurons from the data in the model 1-to-1, and then match the traces through backpropagation and minimizing our loss function, mean squared our loss function. But we also penalize the rank, and how we're doing that is, suppose you have a J matrix connecting all neurons from all regions with each other. Now, each particular connection of an area is, like here, A and C, from A to C, is parameterized by a low-rank matrix consisting of the outer products of the singular vectors M and N, multiplied with their singular values S. Now make use of those singular values by summing them up and penalizing them, adding them to the loss function, multiplying them also with lambda that we find through a grid search. Now, after training and cross-validation, we can plot the distribution over singular values, and we find that indeed most of them got pushed close to 0. So that's good. We can also now take a look at each particular connection between the areas. Here we plot the dimensionality of across and within communication of the particular areas. We use the p ratio of the singular values of those low-rank matrices and see that indeed for V4 and parietal cortex, they share they have low dimensional communications subspaces. The other areas not really, might be due to that we focus on signal and not noise. But also, very interestingly, is that we see a compression of signal towards frontal areas, which might hint to a more abstract representation of our task in the frontal areas. That's also along the lines with the paper, the lower one, in human fMRI, where they see a compression of signal from sensory to association areas. But even cooler now, and that's supposed to be Joao's talk, or also my poster, we now have a generative model of 6 brain regions interacting with each other, and so we can do in silico experiments and do some quasi-causal inferences of their connections and the impact of those. Yes, so if you're interested in this, visit my poster tomorrow or attend his talk tomorrow, do both. And since Joao couldn't promote this, I want to give a quick shout out to Caleb, who is having a poster today. He's also using low-rank RNNs, but fitting single trials of neuropixel and neuromodulators photometry to investigate how neuromodulators shape network dynamics. Thank you.
Hi everyone. I'm going to present my work on enriched experiences, how enriched experience increase the symmetrical connectivity and sparsity in association cortex. This work was done in Bruce McNaughton lab at UC Irvine. So the question I'm primarily interested in asking is, what is the differences in population activity and underlying connectivity between a knowledge-rich and a naive brain? So if you look at work from convolutional neural network or artificial neural network, what happens as the network learn more and more, there is typically an increase in sparsity and orthogonality. And then in a separate line of work, we showed that as there is this increase in orthogonality, the network sort of learns new information much faster. So does this hold for a biological brain? So how do we go and create a knowledge-rich animal, and how do we test this question in animals? So what we did was, we took mice that were relatively younger, so think of them as a teenager equivalent, and made them run on either a control track on the left or an enriched track. And what you can see on the enriched track on the right is these animals were exposed to a lot of different objects and a new obstacle configuration for 10 weeks. And so they're sort of exposed to different sensory modality inputs and so on. And the idea is to sort of send this animal to a university and make them knowledge rich. And then we did high-density silicon probe recording from retrospinal cortex, visual cortex. So today I'm going to mainly show data from retrospinal cortex. And I focus mainly on awake rest and slow wave sleep. That's when memory replay is happening and presumably the brain is sort of exploring its state space. So the first thing I computed was population sparsity using this measure, which basically sort of computes how non-uniform the activity is for different time bins. At a population level. The same thing, you can also compute something called lifetime sparsity, where you can ask, like, given a neuron, how variable its activity is across the entire duration of recording. So what we noticed, so here I'm showing enriched, I'm showing lifetime sparsity for 15-hour-long recording chunked in 3 hours bin. The greenish hue is the enriched animal, and then the black grayish is the control track animal. And so what we observed was enriched animal generally had higher sparsity as compared to the control. And so there was a general trend of enriched animals showing higher sparsity, and this was more prominent in the deeper layers of the retrospinal cortex. Same for lifetime sparsity, there's just more sparsity in the enriched animal. And the next thing I computed was cosine distance, which is, think of it as a measure of orthogonality. So again, enriched animal brain, the neural activity was much more further apart. So these are the changes in population activity. Next we asked, well, what about the underlying connectivity? So what one can do is, given this pair of recording, you can compute cross-correlograms. So if you have a monosynaptic excitatory connection, there's a peak between 1 to 4 milliseconds, so that's EE1. If it's a reciprocal connection, you have peaks on both sides. If it's an inhibitory connection, there's usually a dip in cross-correlogram. So what we noticed was, in enriched animal, there's an increase in reciprocal connectivity, which is EE2, even though the total number of excitatory connection is similar. And then there is an increase in inhibitory connection. So we modeled this, and we used this model from Claudia Klopat's lab, and the idea is that when the brain is operating in this high inhibition regime, what it can do is, it might keep the weight changes restricted to the cell assembly that were used in learning or whatever novel experience, and maybe that's what sort of leads to this increase in reciprocal connectivity and so on. So just to conclude, I showed that there's an increase in sparsity and orthogonality, and my thinking about that, given a lot of manifold people here, is that maybe as we get knowledge rich, there's this sort of increase in sparsity to sort of encode for these large repertoire of experiences that the enriched brain is learning. And then there is this increase, there's this increase in orthogonality, which sort of is, pushing the representation further away. And then these changes are brought about by the underlying changes in connectivity, such as increase in this reciprocal or recurrent connectivity and increase in inhibitory connection. With that, I would like to thank everyone, and feel free to ask me any question if you have. Thank you.
Right. Okay, let's try this. Right, so I'm presenting a project that is termed Algorithmic Representations in the Human Brain that underlie schema generalization. And what I'm referring to when I am talking about schema generalization, I think I need to now always say next slide, right? Yes, okay. Is the following. This is my first time here at Neuromonster in Split, and I'm assuming that none of you has actually been at Neuromonster in Split because it's the first time here. But because all of us have a schema of how conferences in general work, we had no problem understanding what's going on. A conference schema, or a structure of actions, next slide. Okay, this is going to be a next sequence now. Goes a little bit like this. A speaker comes up to the stage. Wow, this is not smooth at all. Next. They start with the introduction. They dive deeper into their results. And then in the end, they might or might not take some questions before the next speaker comes on. Okay, this is now 4 slides in. Can you go lower, please? Yeah. Yes, more. Yes. Okay, before the next speaker comes on. And because we've learned such an abstract structure of how schemas work in general, we can combine it with the current context and have no problem of behaving here. Now, if we think about how the brain might represent such an abstract schema or knowledge plan derived from such a schema, we might think about 2 different representations. 2 slides, please. One representation is a factorization of the abstract state that we're currently in. For example, I'm giving my introduction right now, and combining it with the current context in Split. Next slide, please. Now, it's a bit hard to study going to conferences in a lab environment, so we opted for a slightly simpler schema task, next, which is the ABCDA schema, which we were able to teach both rodents and humans that were going in the fMRI scanner, but also epilepsy patients who had EEG electrodes implanted. The schema is much simpler. Here, subjects have to find 4 different rewards on a spatial trajectory, a 3x3 grid, and have to plan where they want to go. Once they found the 4th reward, D, they have to go back to the first one. And a different context in this ABCD schema, next slide, is simply a different configuration of these 4 rewards. And because both mice and humans were able to generalize the ABCDA schema, they were going immediately back to the location where they found the first reward once they found reward D in the context number 2. Now, next slide. It might also, oh, one more, sorry. It might be, next. Okay. Thank you. It might be easier for you to understand what I meant earlier when I talked about the factorized representation of the abstract state of the schema together with the current context. Because the current context here is simply where am I on a spatial trajectory, which we all know is represented in place cells in the hippocampus. An alternative way of representing such a knowledge plan, next slide, is something like this. We could imagine that there are neurons that don't only represent where we are right now, but also where we are in all steps in the future. This sort of representation doesn't only tell us, or it tells us also what the structure of the schema is, because we infer from the schema structure how long it will take us until the next speaker comes on, give the introduction, and so on. The cool thing about this structured memory buffer way of representing an action plan is that colleagues in our lab, next slide, actually found this representation in rodents that were solving this ABCD task when they were recording from their prefrontal cortex. Now, what I did here in this project, next, is I looked at the firing rules that they extracted from the neurons they recorded in prefrontal cortex in mice, simulated the neurons, next, based on the trajectories that the mice were taking, and then tested using an RSA whether I could indeed predict the neural data they were recording from mice. Knowing that my SMB model of neurons was valid like that, next slide please, I then took this simulation of neurons and asked whether there was some area in the human brain that also represented the ABCD schema in a similar way. Next slide. And doing a searchlight procedure, which basically means testing every single region in the brain, we indeed found that the analogous region to the region where we recorded from in the mice, medial prefrontal cortex, also represents the ABCD schema task in the same structured memory buffer-like way. Additionally, next, we also found that this abstract ABCDA schema representation, independent of context, was represented in entorhinal cortex, which nicely fits the literature of representing abstract structure independent of context. Next slide. Next, we were also interested in knowing how this structured memory buffer-like action plan actually gets to medial prefrontal cortex. And for that, we both looked at fMRI data, this is a video which is probably not going to play, as well as epilepsy patients that had IEGs implanted. Next. We found that when subjects had all information about the current context, this action plan representations arose in medial prefrontal cortex exactly once they were starting to plan. Next slide. And lastly, that this planning phase correlates with increased sharp wave ripple rate, which is a signature of replay. So our hypothesis is that potentially replay might play a role in getting the information of the current context, so medial prefrontal cortex, in order to load this action plan. And with that, if you're interested in more, next slide, come to my poster and talk to me. Thank you.
Cool, so now for something very similar, but different in flavor and with slides. Yes, I'm going to tell you about, my name is Alon, I also work with Tim, same lab as Svenja. And I'm going to tell you about work I did together with Tim Behrens and Mona Garvett about how the brain represents information for fast inference and generalization. Right, so here's a family tree of 3 generations, and if I now add a new node here, and the only thing I tell you is that Emily is the child of Bob, you'll be able to immediately infer all other relationships of Emily to everybody else in the family. And you can do that because you know some stuff about families, about family trees, and about their structure. So how should the brain represent such information in a way that is useful for this kind of inference? One way of doing so, which is particularly powerful, as shown by James Whittington, is to represent the structure of this problem explicitly, in a way that is divorced from any particularities or sensory details. And here in this task, in this project, what we do is look for such representation using fMRI in humans. Right, so do I have a laser pointer here? Yes, good. Here's what we do. We train subjects on associating arbitrary pairs of stimuli. And the associations come from a graph with this structure. If you're connected on a graph, then you're associated. Importantly, participants never see the full graph. They only experience it pairwise. Now, the next day, we train them on the exact, on a similar graph with the same images, but now the distribution of the images on the graph is orthogonal to the first graph. As you can see, the structure, and this is crucial, the structure of the graphs is exactly the same. So here's the timeline. We trained them for 3 days, and then on day 4, we scan them, and then we scan them again 1 to 6 days later. And we're going to talk about differences between these two sessions in their presentations. Right, so what happens in the scanner? They see things like this. They see, the resolution is a bit crap, but never mind. They see an image in a context. We always tell them which map they're on, blue or red, with the background color. So stuff like that. And we're going to use an approach called repetition suppression, fMRI repetition suppression, which assumes that the closer the current stimulus is to the previous stimulus in the representational space encoded in a particular brain region, the more suppression of board activity you'll see. So it's easy to think about that in visual cortex. If I show you 2 exact same images of this chair, you'll see to the second image, you'll see suppression throughout the visual hierarchy. But if I show you an image of this chair and then a rotated image of this chair with some different lighting, you'll get suppression to the second image only in higher visual area, which are invariant to these kind of things. So I'm going to split my trials into 2 types: state trials where the context stays the same, red to red or blue to blue, and switch trials where the context to the previous trial has changed. And we're first going to focus on state trials where we can ask, when the brain, do we get a suppression effect that corresponds to the currently relevant map? Here it's blue. So here, where do I get a suppressed effect to the ear? And the suppression should be stronger, so less activity, if it came after the shirt, which is close to it on the graph, than if it came after the motorbike, which is further away. And we can play the same trick with the irrelevant map, which has different distances. And the nice thing is that because these distances are orthogonal to each other, because we built that this way, we can look for regions that encode both the relevant and the irrelevant maps simultaneously at the same time. And when we do that, we get a nice blob in medial temporal lobe across both sessions, and that fits nicely with some previous literature, and I can talk about that with anybody who's interested more in my poster. And now to our main question. So can we find an explicit representation of the abstracted graph structure? So here we're going to focus on switch trials, and now we're going to define our distances to use for repetition suppression. We're going to use this abstracted graph. So here the distance between the ring on the blue graph and the bed on the red graph will take where the ring is on the blue graph, that's here, and we'll take where the bed is on the red graph, that's here, and the distance should be 3. So this is actually encoding the location of the image on this abstracted graph, not on any particular graph, on whatever graph was relevant at the time. So when we look for brain areas that have suppression with this kind of distances, we get medial prefrontal cortex, very strong effect, same region as Svenja got, and actually this comes only from session 2, and you can see the map of the difference between sessions in the red-yellow map. So this is reminiscent of stuff that Jay McClelland came up with about consolidation mechanism that happened with time. Finally, we replicate this analysis using a completely independent pipeline, RSA, and I can tell you about that more in my poster, but because it's a completely independent replication, we think it's believable. And really, finally, this fits really well with stuff in the literature about schemas and about grid-like signals. So always the strongest grid-like signal in 2D tasks are in this medial prefrontal cortex area, which supposedly encode the structure of 2D tasks. And we can chat more about that in my poster. Thanks.
Yeah, to present the work that I've been doing at the Center for Neural Dynamics and MEG at the University of Tübingen on neural interactions across cortical layers. So this is the session on neural data. So I would like to start by describing the type of data that I've been working with in this project, which is laminar electrophysiology data, in which we record the activity of neural populations across all of the cortical layer, like all of the layers within the cortical column, so that in the end, our data has both a temporal as well as a spatial dimension over which it evolves. And we do this because we are interested in how different layers in the cortex talk to each other and exchange information. And we would like to measure this by computing some measure of the statistical relationships between the neural signals. And that's because while on the anatomical level, we have a fairly good understanding of what the structure of the circuit is, at the functional level, especially when animals are behaving, the picture is not so clear. And so the major question that we try to address is whether by taking measures of the functional connectivity on this spatiotemporal signal, is it possible to retrieve some anatomical features of the underlying neuronal circuit. Now, there are many measures for functional connectivity, but by far one of the more popular ones in neuroscience is Granger causality, which is just model comparison method based on linear regression that asks how much better can I predict a given time series if I include in my regressors the past of another time series. And we can ask this in both directions, so we can also quantify the asymmetry in the interaction, meaning which signal drives which more strongly. It's a very popular method, but we know it has some statistical limitations, and what we argue is that in the kinds of recordings that we are dealing with, these limitations are made worse, they're aggravated. And so another question we try to address is whether Granger causality really is a valid measure for estimating connectivity between cortical layers. So to address these points, I went and analyzed part of a much larger data set collected in our lab of multi-area laminar recordings in macaque monkeys. I mostly focused on the prefrontal areas from which I extracted the local field potentials at every channel along the cortical column. And with this, I compute the Granger, like the pairwise Granger causality between each pair of these channels, so that in the end, I get these kinds of interaction matrices where you have basically channels on both sides, and this gives you an idea of how every channel in the probe is predictive of every other channel in the same probe. So we are within area. There is no across-area interactions in this work. And one feature that I'd like to point out is that the upper triangular component of this matrix reflects downward projecting interactions, so from superficial to deep layers, whereas the lower triangular component goes in the other direction, so from deep to superficial layers. And I point this out because I think it's evident by just looking at this example matrix that the lower triangular part is has a much stronger Granger causality signal than in the other direction, implying that in this data there is a very strong upward biased asymmetry along the cortical column, which is something that we find across all of the recordings that we have, across all of the prefrontal areas that we've looked at. And looking in the literature, there's a few studies that have done similar kinds of analysis, and they also report this asymmetry, yet nobody really tries to explain where is it coming from. So that's kind of where we try to come in. And we try to explain this asymmetry using a source mixing model in which we imagine that there are one or more independent neural populations without any interactions that project their activity onto a set of measuring channels with some noise. By analyzing this model and running simulations on it, we can show that an asymmetry like the one in our data can actually is guaranteed to exist even if there are no actual underlying interactions in the generative model, which means that at least partially our data could reflect this kind of noise-driven asymmetry rather than through interaction. So to get a better understanding of how our data is affected by this, we apply the matrix factorization method to these laminar Granger causality matrices that decomposes them into a sum of non-negative components. When we apply this to simulated data where we control the position and the mixing of the populations, we see that the components retrieved align very well with the mixing coefficient that we impose by design. And when we then go back to the data and apply it there, we also see spatially well-separated components where the peaks of these components align well with the 3 major laminar populations estimated with other neurophysiological methods. So in conclusion, we can say that yes, Granger causality can give us information about some structure of the circuit, but if we really want to use it to tease apart the interactions, we need to account for the source mixing effects that might obscure through interaction. So with that, I would like to thank Marcus and Konsti for supervising the project and the rest of the team, as well as you, the audience, for the attention. Thank you.